Hello students, let us continue our chapter Human Digestive System, and today we'll be talking about the finger-like projection that is present in the inner walls of the intestine, that is villi. What is villi? Villi is nothing but the finger-like projection which we can find in the internal wall of the small intestine. Villi is helpful in absorbing the digested food. If we see the internal structure of villi, that comprises of vein, artery and lymphatic duct, together forms leg tail. Then we will be talking about bile. As I said, that bile is produced in the liver. Bile is produced in the liver and it is being stored in the gallbladder. At the time of digestion, that bile is released in the duodenum part of the small intestine. So, in the liver, when the bile is produced by the help of hepatic duct from the liver, is transferred to the gallbladder. Then, gallbladder consists of a duct which is known as cystic duct, which is present here, where I have pointed, and then together hepatic duct and cystic duct together they form common bile duct, by the help of which they secrete the bile in the intestine, small intestine, that is duodenum part of the small intestine. So this is how the liver secretes the bile in the small intestine. Now let us see what is the function of bile in the work of digestion. First of all, it helps in the breakdown of fat into small droplets of fat, what we can say it as emulsion of fats or emulsified fats. Secondly, by in the presence of sodium bicarbonate, bile helps in converting acidic chyme into alkaline chyme. We remember that the food is coming from the stomach. In the stomach, acid is present, that is hydrochloric acid. Now this hydrochloric acid, when it gets mixed with the partially digested food, it forms acidic chyme. When it is transferred to the small intestine, first the work of the bile is to convert it into alkaline chyme so that the further reaction may take place. Now let us talk about the pancreatic juice. As we said that the pancreatic juice is also secreted in the small intestine. Pancreatic juice also contains certain enzyme, that is trypsin, amylase and std-ix. Trypsin is activated. Trypsinogen is in inactive form. Trypsin is in active form. So trypsinogen is converted into trypsin by the help of the enzyme, that is enterokinase, and then trypsin can act upon the leftover proteins and peptides to convert it into smaller peptides and amino acids. Then present amylase. Amylase helps in the conversion of leftover starch to form maltose. And std-ix helps in the conversion of emulsified fat to fatty acids and glycerol. So these are the enzymes which we can find in pancreatic juice that helps in the digestion of the leftover substances into their end products. Now let us talk about the intestinal juice, that too is secreted by the walls of the intestine, which contains certain enzymes which are present here. Like peptides, the leftover peptides is again acted upon by the iripisin or peptidase to form it into amino acid. Maltose acted upon by the enzyme maltase to form glucose. Sucrose is converted into glucose and fructose by the help of sucrase. Lactose is converted into glucose plus galactose by the help of lactase. Emulsified fat is converted into fatty acids and glycerol by the help of lipase. So these all are the intestinal enzymes which are secreted by the walls of the intestine along with the intestinal juice, and helps in the conversion of the leftover substances of the food to the final product. So these are the final products, and now these substances are ready to be absorbed by the intestinal wall. Then let us talk about large intestine. So small intestine was the last site of the digestion of the food, and now comes the large intestine where the absorption of water may occur and a few absorption of food may take place, and as it doesn't contain any enzyme, so no further digestion takes place here. Now the large intestine is again divided into three parts, that is cecum, colon and rectum. Cecum is the part where the small intestine and the large intestine joins, that is a junction of small and large intestine. Then comes, and the cecum is provided with appendix which is present here, you can see here. Then comes colon, comes ascending colon, transverse colon and descending colon, where further digestion of water absorption of water takes place. And then the last structure comes rectum, where the temporary storage of the feces occurs, and when at the time of elimination the rectum they contract and the feces is removed from our body. So this is the structure of large intestine. So the function of large intestine: secretes no enzymes, so no digestion. It absorbs much water and mainly consists of undigested food. It forms a semi-solid substance, that is feces. The expulsion of undigested remains of the food from the alimentary canal is called defecation. Let us now come to the last step of the digestive system, that is assimilation. Assimilation, it is a conversion of the absorbed digested food into body material. Like glucose, as the starch is finally broken down into glucose, then glucose is the excess of glucose is converted into glycogen, and then it is being stored temporarily in the liver. When again the glucose is required by the body, then the glycogen is converted into glucose, and then the glucose is utilized by our body to produce energy. So glucose is converted into glycogen, which is being stored in our body. Amino acids cannot be stored, so excess of amino acids are broken down in the liver by the process, that is deamination, in which the nitrogenous waste in the form of urea for excretion. So that forms a nitrogenous waste and that forms a urea, and that is the process of excretion. Fatty acids and glycerol, sometimes they gets deposited under the skin and provides energy to the body. So these are the finally assimilated products or the food that that is converted finally into body material. So in our next video, we'll be talking about certain experiments of digestion.